Hi there.
Welcome to YouTube Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for the university, or for pharmaceutical advertisers, and the president of Biotech Without Borders, a nonprofit based in NYC, dedicating to the public with the tools of biotech.
My name is Faz, and in the previous life, I got my PhD in microbiology from Imperial College London, where, amongst other things, I was making flesh-eating bacteria glow in the dark. And currently, I'm working as an editor for an academic journal.
Every week we meet to talk about microbiology, and today it's our deep dive week, where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, and we encourage our audience to leave us questions and comments.
And next week, if you join us, will be our news week, so where we survey a bunch of different articles and choose which article we'll be looking for the next deep dive. So make sure you subscribe to satisfy your microbiology curiosities.
And you can follow along with the papers we discuss on either week in our shared Zotero in the Zotero library in the doobly-doo below.
And we want to hear from you, so please use the comments to tweet us using the hashtag MicroTWJC hashtag. And
So today we have something that we chose from our first non-COVID paper in a really long time. We looked at like top cited papers last week, and this was number one.
yeah, so the title of this paper is A Deep Learning Approach to Antibiotic Discovery. So we hope there'll be some deep learning happening today.
Yeah, the first slide of this we have is like this question that they're asking. I guess AI is such a popular sort of object in our world these days, and we want to use it for every problem under the sun. But of course, like that's not super straightforward. So yeah, the big question is, can we use this AI technology to find new antibiotics?
Yeah. So why would we need new antibiotics? I mean, there are plenty of reasons. The fact that we're running out. Yeah, I mean,
Yeah.
historically antibiotics were created by bacteria as weapons used against other bacteria, and we kind of stole it from them and used it for our own uses. And
Yeah,
yeah,
there are tons of different types of antibiotics out there. I think people have like a timeline of antibiotics. There's a golden era of antibiotic discovery, and we're sort of past the golden era. When people try to find new antibiotics now, sometimes they find basically examples of old ones, and like that doesn't work because if the bacteria has become resistant to it, like they have the same mechanisms if you're finding antibiotics in the same class, right? So there's this big push, right? Like you can characterize antibiotics by like the types of things they disrupt. So like in this figure, right, you can see there's like cell wall synthesis, there's nucleic acid synthesis, there's protein synthesis. Lots of things go into making a bacteria tick, and antibiotics could fall into a class based on what they disrupt.
yeah, and actually the discovery of antibiotics has kind of slowed down a lot recently because in the olden days it used to be as easy as finding a new species of bacteria and finding that it clears out something from a plate, and then you've got a new bacterial product. But then we kind of got all those low hanging fruit, and now it's getting much harder to find new antibiotics.
Right, right. And so on to the next slide of our intro.
Yeah.
This is a bit of a foreshadowing because it comes out of nowhere, but as I said, many different ways of having bacteria live and die, right? And an important thing that bacteria need to live is something called the proton motive force. It runs things like ATP synthesis, transporting solutes, so like getting sugars into the cells, and it can also be linked to motility. And so that's just one of the many things that go into making a bacteria live. And yeah, it's a foreshadowing because if we break it, we could kill a bacteria.
Well, I mean, it's important because antibiotics target the bacterial metabolism. So the idea of a good antibiotic is something that will target bacteria that doesn't target humans. So we want to look for processes like this that we see in bacteria that aren't necessarily found in humans, so we can target them specifically.
Right. Yeah, and I guess that's a big thing about drug development in general, right? This language of like targeting a system, right? Like you learn enough biology about something and you say, oh, now I want a molecule that goes in, right, and can like break this thing because we don't want it to happen anymore. Yeah, I don't know. That's just like maybe an interesting piece of language. So in our next slide, we have this crazy picture of some of the ways you find drugs. So large pharmaceutical companies basically make libraries, combinatorial libraries sometimes, or sometimes it's like molecules that they think are going to work. And so that image A is like one of these libraries. So imagine this room just full of plates where every well has a different molecule in it. And there's some idea of like how that molecule could be effective, but you don't really know what's in it. They made all the molecules, and the idea is that if you test them all, you'll find something, the needle in the haystack, that works in the way that you want it to.
Yeah. So they've got like, and yeah, this is essentially like the equivalent. Most testing is equivalent of throwing spaghetti against the wall and trying to see what sticks,
Yeah,
essentially. So
totally.
testing.
And that's why they have robots do this sort of stuff, right? Like, it's not something that it sounds weird, right? Like, there's not a lot of knowledge about the pathways necessarily that can go into this. I guess it's all about assay development, right? Like you do one assay is the idea, and you test all of those molecules, and you'll get things that work in that test. And how you set up that assay is really important. That's the stuff that uses, I guess, the brains. That's like the brains of the thing, like figuring out the right assay. But then after you know the test, you just kind of brute force it. That's drug discovery right now,
Yeah,
uh, in a nutshell.
because we are so far from understanding every single thing that goes on in a bacterial cell that we are kind of stuck trying to like throw random chemicals at it and see whether it makes them stop growing, and
Yeah,
hopefully just through like testing every single thing in existence, we hope we'll find something that'll work.
I mean, I guess it is like it. I like to think it's super random. They do also do some things like libraries might be very soluble, right? They optimize other things, right? Like that you can know from the chemical principles, right? So the library may
Right.
have things that like they already know how to make, right? Or that they know like isn't toxic, right? Maybe toxicity is important, right? Like they have libraries that are not like not toxic to other things, but the speed at which you can design molecules is certainly outstripped by right, certainly outstrips the speed in which you could make molecules, right, or even test them. So that puts us in this sort of tricky scenario. But yeah, this image here is an assay, right, that you could use to test for antibiotics, right? It's called minimum inhibitory concentration, and so the idea is you dilute your compound of interest down, and then you see at which concentration is the lowest that it can be that still stops the bacteria from growing.
Right, and that's like been a classic for antibiotics for 50 years now. Um,
Mhm.
so now just looking at the whole pipeline, because there is a lot of chemical diversity out there, but there are like certain limitations. I think you've mentioned before, like kind of something has to be able to dissolve in order for it to actually get absorbed into the human body to be used for treatments. But there are other sorts of things like things that are incredibly dangerous. Don't want to put in the human body, like something with like that produces cyanide or something like that. Just um,
Yeah, there are things that are they might like, or they might even just hit pathways that you don't want to put in the human body. Like you don't know, right? Like if you're working from a huge amount of just unknown small molecules, there's all sorts of stuff in there. And yeah, only through testing can you really get it. And here in this graph, they actually give us like an estimation of like how much you could reasonably think to test, and they give it at like 10 to the 6. So that's like a million, right? In the
yeah,
millions range. They can test millions of things, but more than that, tens of millions, hundreds of millions, billions, trillions. I mean, that just becomes really unfeasible to test every single thing.
right. So the one other way to like figure out how to do this is to try and teach a molecule to figure out what are you likely drug what could make a chemical an antibiotic. Uh, because
Yeah,
the stuff
so
for
this is the computer stuff, right? Like this is what they call in silico approaches.
mm,
Yeah, because computers, I guess they don't need to be tied right into the physical experimentation. They might see patterns, and those patterns are going to help us narrow down that funnel.
yeah, and computers are kind of hypothesis agnostic. They don't come with any biases or well, we if they do come with biases, they're ones that we put in or that we can kind of hopefully discover later. But
Mhm.
the basic theory is that they can go empirically off the data and try and figure out like what properties there are without trying, and they could potentially find links that we wouldn't occur to us because of the
Yeah,
way we're taught and the way we learn about chemistry.
Totally. I mean, they also just have like they can just systematically go through everything, right? Like they are taking the same criteria and looking at each thing. And so this has been done before, right? This idea isn't new, right? There's this there's this concept called a quantitative structure activity relationship, right? And that's basically saying that there are features in a molecule, structures in a molecule that map onto some sort of activity. And if you could figure out, right, that's like the golden thing. If you can figure out what structures map onto what activities, now you can do prediction, right? Because you can figure out, oh yeah, like when this bond is a nitrogen and it's connected to like a group that has this cluster of a ring and like whatever, that's an antibiotic. Then now you have like the key, right? The key to understanding all this stuff. You know what to ignore and what not to. But we're kind of it's difficult to do this because there's like you can make molecules that are of all shapes and sizes. People don't have a good concept of what an antibiotic is, and I think this is where the authors are thinking along the lines and papers before have thought along the lines of, well, maybe we can train, right, this machine learning technique can train a computer to get this information.
Yeah. So they've got like all these list of parameters down here, like things like atomic weight. Like, I mean, you could read the list below and all look at in the actual paper. But yeah.
It's all chemistry stuff. This is all the formal ways you can describe a molecule from the chemistry standpoint.
Yeah. And so how do you chug that into a computer so the computer can understand how each atom bonds together, how those bonds not only affect the atom but the atoms next to it, and then atoms, and the kind of how it changes across the whole molecule.
Yeah. I mean, I think part of this we can also think about as it's like we've talked about like multi-dimensional data, right? Like each one of these, right, is an axis on a graph,
Yes.
right? A really complicated graph that branches out in like all these different directions, and every molecule has its unique pattern that sits in there, but some of the features of that graph are important and some of them are not. And the first step would be getting that whole feature map into a computer in a way that can be manipulated, I guess, and simplified if necessary.
So yeah, basically, so to get it to a computer, we almost have to like look at so you traditionally used to like break it down to functional groups. Go, oh, this functional group will bond to this, and but giving that to a computer, the computer would look at say that group and wonder what does this do, and then build out by figuring out what relationships is. So what are the other atoms that are bonded to it, and how that might affect it, and then each like atom now. So trying to generate an idea so that each atom kind of knows what's in the other structure just by the basis of the bonding and the whole structure. So I think there's this idea of like. I mean, this goes into a physics which is a bit beyond me, but I think the idea of like some information being contained for all the structure in one part of it, because every part of the molecule is affected by the other part. So there is even like in chemistry, there is some information of the other parts of the molecule stored in there. So that's
Yeah, totally. I mean, that's like I don't know if people remember this part of chemistry. I'm not sure if you remember this part of chemistry files, but like electronegativity, electropositivity, right? Like molecules are like these clouds of electrons. If you have charges in different regions of those molecules, it can pull those electrons away, right? And that's changing the chemical properties of things, right? Like these partial charges that sit in different areas, that's all relevant to how the molecule works, how it binds to different things. And this way of like systematically going through the molecule and saying this region is attached to this region, it's a map, right? It's
right.
or a fingerprint. And this is like the most basic way in which you could encode information into a molecule.
Yeah, which brings us to our next slide. So deep convolutional neural networks combine these molecular features with like the various like target properties, um, and then they like iterate over them to get like an idea of what it looks like overall. So
Yeah. Okay. So this is um, this figure is partially like the computer learning that goes behind it, right? So the idea is that right, like each of those bars underneath it, right? That's the identity of the molecule, right? That's that table, right, that we showed earlier that says like, oh, that maybe white, right, means like is it hydrogen or is it carbon, right? Like those are each a description. Everything has a description, but computers are really good at transforming information, right? Like that's a big part of simplifying it, right? Like if you just have a huge table of every molecule and its properties, that's one way to represent it. But you can also, as you were saying before, they have some relationship to each other, right? So you can kind of mix, you can say that this is dependent on having a different table beside it, right? Like if the table is beside it, they have an effect on each other. And essentially that would make like a different color, right? Like if you can see from 1 to 2, right? Like at first they have these short
Hmm.
tables, right, that only have the properties. But in number 2, once you convolute it a little bit, right, once you take the information from around it and multiply it together, the table becomes a bit longer and it looks a bit different. It's still a unique fingerprint for that particular area, but now it's being impacted by the information around it. And you can iterate that over and over again, right? To essentially, and you can group, right? You can group those tables in different ways, right? They have graph pool and graph gather. You can group those in different ways, and essentially you are compressing the information, right, that you get from the whole molecule into a single like statement, a single fingerprint, where everything isn't, it's not, it's abstract now, right? It's not related to the physical properties, right? Only through like these mathematical transformations does it become related back to like what the molecule is.
Right, right. Uh,
Yeah.
so yeah, this is a part of the paper that I, as like a microbiologist, was very difficult for me to get across. I think you've kind of explained that a bit better. That kind of takes all that information, compresses it down to create this fingerprint that basically is derived from all these molecular features and how they relate to each other.
That's right. That's right. And the big thing in machine learning is weights,
Hmm.
right? Like, but which features are important? So when we're thinking about what does the machine learn in all of this, what it's trying to learn is how to weight each of these like features so that the end fingerprint that comes out is very clear in associating structure to function. Right? So you would, this is the way that it learns what it is, but of course you have to supply like the definition, right? You have like a training set that says all of these molecules that have this particular fingerprint, right, they associate with this phenotype, right, this particular activity. And the computer's job is the computer's work is to say like what's the contribution of all these different pieces of data, and like that's like, you could play around with those knobs forever, right, until you find the right resolution between groups.
Right.
Computers do that easily because that's what they are, right? Like they can infinitely go through and just like adjust everything a little bit to find out what makes the perfect match.
Right. And then they got this thing called a direct message passing neural network. Again, this is something that I'm not too familiar with because I think it's like they describe it as a way to just to take convert like molecule structures, which are like graphs, and then push them through a neural network.
Yeah, so I mean this is like these two things are related, right? They are using these the direct message passing neural networks, the DMPNN. This is a convolutional neural network. This is a convolutional neural network, right? Like it's just right, because it actually it's doing exactly what I said, right? It takes information from around and it compresses it down. Like if you think about, this is a graph that comes from, well, they actually put it in their graphical
Hmm.
abstract, right? It comes from a previous publication, right, that does the same thing. In essence, those 3 dots, I don't think they do a good job here, but like those 3 dots are supposed to be like the information, right, contained in a bond, right? That's the stuff that you can read off of, that you could read off of chemistry textbook,
Right.
right? Or like from looking at structure, you can categorize things, and they're saying that as we were saying previously, that string is influenced by strings around it, so they concatenate them together to make a new one, right? And you can iterate that over and over again. And so what was found in the past, this is previous literature, right, is that these types of networks are really good at categorizing things, more so than other categories that exist. So that's table 8 at the side, right? Like there are other data sets that exist there, right? I guess they're calling them RF on Morgan, FFN on Morgan, FF, right? Like those are different algorithms that can go through it, and you can see that they show that their DMPNN algorithm is better, right, in these different cases. So it's worse in some, it's worse in a few cases, right? It's the same in some cases, and it's better in others. So this is the advance, I guess, in computer science that tips off people to say, I want to use this algorithm, right, to solve a problem. Because it performs well in these tasks. And maybe I should explain, like, when we say tasks, right, it's machine learning, right? So you have a training set and you have a question set, right? The training set has everything explained for you. It has all the molecules and what they do. You train your algorithm on that training set, and then you say, now test yourself on this other unknown set, right? And that's how these algorithms are evaluated, how well they do with a piece of training information to then classify like a test.
Yeah, so had this. They basically built this into the pipeline now. So now they show this training set to this AI, and the AI is looking at all the chemical properties and figuring out, okay, this is what an antibiotic looks like, and then setting out on this vast library of other things, and then getting it to then figure out that, okay, what from this library looks similar to what I've learned from my training data set.
Mm-hmm. Mm-hmm. Yep, absolutely. And the way that the computer is getting there again is by like changing the weights of how it thinks different features are important,
Right.
right? And those features are ultimately all being mashed together by maths, right, to give a single it's a single, but it's a vector. It's like a really long string, right, that explains the relationship between all the features of the molecule.
Right.
And I think we have an explanation coming
Yeah,
up.
so this was my attempt at trying to figure out. So essentially, starting with just this one sulfur atom on one end of the molecule, it passes a message to the other kind of atoms next to it to let it know that it exists. And again, we're passing on that like table of information that it has of like this is
Yeah.
all my information about myself. This is my Tinder bio, putting it to the other atoms, and then
Mm-hmm.
those atoms take that information as well as their own information and passing it on to the other atoms, and then it iterates and iterates until all the atoms have passed enough information. But obviously, there's different. So the AI doesn't know what the size of the molecule is coming in. It doesn't know how many iterations it needs to make in order to get a picture of the whole molecule. So that's why there's
Mm-hmm.
this thing called hyperparameter optimization, where it kind of decides how many iterations to use, and so it uses these various reasonings to figure out, okay, how many iterations should I use for this molecule, and kind of then using various clever mathematics to figure out how it should learn. So that's
That's actually empirical. Like the strange thing is that's an empirical measure. Hyperparameter optimization. It actually doesn't. It's not like the computer determines. It doesn't know de novo, right? That like this is how many it should use. It has to measure a metric. It has to measure some metric, and so really it's just does more iterations, right? Does incorporating more information into the model, iterating more, getting more messages, does that make my job of classifying better or worse? And that's how the computer decides,
right.
right? It's very abstract, right? Like there's no knowledge that it's applying from like the chemistry or anything like that. Like all of that becomes abstracted in the computer's eyes. And then maybe this is kind of what you were saying before, right? Like the computer isn't like a it doesn't have knowledge as much as it like is just executing an unbiased set of rules, and as long as we're setting its parameters right, like to say, oh, we want this goal of like being able to distinguish between these things, then it will do who knows what it's doing. Actually, this is the whole point of deep learning, right? Like we don't really know like what mathematical rationalizations it's using to say that one thing should be weighted higher or lower. That is the whole point of deep learning, right? Is that the computer can make that decision because it knows best, because we're just telling the computer, make sure you can distinguish A from B. And the more A's from B's that you can distinguish, the better it is. Just keep doing what you're doing. Keep doing the math you're doing until you get to that point.
Right. So yeah, that's amazing. Another thing that I,
Mm-hmm.
like kind of that the researchers brought up in the paper was that okay, so each in order for each message pass, there needs to be iterations. But the question is like if you have a molecule that's big or complex, like you might have to do quite a lot of iterations, and the computer might decide it's best not to. In which case, you'll get points
Mm-hmm.
where where, like, say, a molecule on one side of, sorry, an atom on one side of the molecule won't necessarily be aware of an atom on the other side of the molecule. So therefore, the
Mm-hmm.
question is, will they still interact, or will they not, or will
Yep.
they miss out something chemical because they've missed out on that extra
Right.
level of.
Or maybe the thing is just how would that information be incorporated at that point, right? Because we've left the computer to do its own thing, and it's made its best decision on what it thinks are the important features to characterize. But we as humans know that there are other things that can influence that, right? And so this is sort of us imposing, saying like, well, what about, right? Like we don't fully trust the computer to make decisions at some level, right? We know that it's going to optimize based on this, but we can bring in our own ideas of what should be in that mix and impose that onto the end result.
So yeah, they added some extra like information or features that you can, but through this other software, I don't know, like kind
Mm-hmm.
of resource called RDKit that they put on top. So
Mm-hmm.
I mean, when they test their model in the paper, they'll use it with or without the RDKit to kind of see whether how much it adds or not. Um,
Mm-hmm. Yeah, and that's just a way of, I guess, shoring up, I guess, shoring up the amount of information that's in there, because we know de novo that probably there will be effects from stuff that's far away, and so we're just like enforcing that into the algorithm here.
yeah, and next slide. So
Right. So a little bit of a summary again, right, of everything that we've seen. And like I just pulled it out. It's like it's pulled again from like all sorts of places, right? Like the table in the back, that's some of the RDKit like features. Then like there's the atom features, right? We know those. We know that that has to be parsed through this message parsing, and like I highlighted this figure again because that really is like the best way to think about this, right? Is that like there are tables represented by those bars with different colors, with different shades in them, right? Those tables are what we observe about the molecules and the bonds. And through this message passing, they're able to concatenate those tables together, right, to make more complicated tables that now, like, they're still all related, but it's getting abstract. We're moving to abstract land, right? And you keep doing that over and over again. And then you ask the computer, okay, so now based on what you saw, right, and this new string that came out, right, what is the right way to weight everything in order to give us the resolution between molecules that do have activity and molecules that don't? And through once that whole mathematical structure is in place, you optimize it, as you were saying, hyperparameter optimization, right? Is just like saying tweaking, I guess, more like higher level things in the algorithm. And then you can also ensemble things together. You can also like do groups of, you can have the machine run. I think we have something to describe this later as well, but you can have the machine run over and over again on smaller sets or distinct sets, and hopefully they come out to the same answer, right? You average those together and you're like, that's better than just training a single thing.
yeah, so essentially you get to this point where our AI now understands the molecule. It has an understanding of what the bonds do, how its structure works. And then, so where do we take that? So now we have to the molecule has to figure out, okay, I know how this works, but how does this other molecule work? And we can help it along with our training set. So, so,
I think this is a little bit of a like the training set already had to go into the model,
yeah,
right? So in some ways we just this is like we're skipping into, or we sort of skip this one part, right? Is that that's all fine that we can have a machine absorb everything from a molecule and figure out some mathematical fingerprint. But I kept saying that like it's about weights though,
mm,
right? And those weights are determined based on its ability to distinguish between things that are and things that aren't antibiotics. So this data that they pick up in figure 2a, right, is that training
yeah,
data, right? This is molecules that they tested that they say this is an antibiotic, this isn't an antibiotic. And so the computer can do all the fingerprinting stuff, right, and all those convolutions, but all that is worth nothing if you can't tie it to a phenotype. And the way that you tie things to a phenotype is through the training set, right?
yeah. And this like set was essentially it was taken from a library of 1,760 FDA-approved drugs and 800 other natural products. So the thing that they tried to look for here is differences in structure, so chemical diversity. They want to make sure they found as many different structures as possible, because otherwise, if they're all very similar, then you just hemmed down in this alley of just one kind of
Right.
chemical structure. They want to be able to, the thing they really want to do in this paper is find an antibiotic with a structure completely different from what we've seen before, to find an activity
Hmm.
that's completely different. So
Hmm.
they pass them through it.
I mean, it's biases, right?
Yeah.
Like for example, like if you gave a data set that only had a certain type of molecule, right? Then when the computer took that in, right, and they made all these strange fingerprints, right, through their convolutional neural network, but through the directed message passing, that those fingerprints, they would say, oh, it's really easy. All of these molecules that we've seen, right? Like all these antibiotics are all the same, right? Like this is the thought experiment. If we put only one class of antibiotics in as a training
Hmm.
set, right? Then the computer would be really good at finding that one class of antibiotics. But that's not what we want the computer to find, right? We want the computer to find new antibiotics. So we kind of take this shotgun approach in training to say, well, like what about all this other stuff, right? And some of these molecules in the approved drugs, some of them are antibiotics, some of them are not,
Hmm.
right? And I think that that's an important thing. I think that's why it's nice that they put 800 natural products in it as well, because some of those are, right,
Hmm.
like there's even less known about those things, right? Some of those are just things, right? Things that they can make, things that they found in other libraries. And that's ensuring that the computer doesn't get biased down a certain path, because it's very easy to train. Like the computer, again, doesn't know. It only knows what we show
Hmm.
it, right? The examples that we've shown. And if it gets trained on something that's too similar, it won't give us the results that we want, this novel result. It'll give us results, and they'll be good, right? Like it will find antibiotics. It will just find the ones that we don't want to find. The whole point of going through this exercise, right?
Right. Yeah, and so in these graphs you kind of see in red are the inhibited. So they measure things by optical density. So that's basically shining a laser beam and seeing where the bacteria disappear. And the ones that disappear are very low in the optical density range, and they're highlighted in red. So they
Yeah,
found,
they never grow, right? They never
yeah,
grow,
they
and things will start absorbing laser light when they grow.
yeah. So like it'll be yeah. So they have an initial dose, and it'll just decrease a lot. So you will get some left over in the background because there's still like some stuff left over from bacterial popping, but um, popping is not a technical term, but it's
Lysis.
lysis. Yeah, lysis. That's right. Um, but yeah. So moving on to Fig S1b, they rank all of them in terms of like which ones uh, were the best at lysing the E. coli, and which ones were the worst. And you've got this.
But not lysis, right? Like this is MIC. This is
Oh,
inhibition,
right. That's right.
not growing. Not growing. Yeah.
I mean, yeah, I get, yeah. So this is based on relative growth rate. So before it was optical density. So yeah, um, yeah. So they rank them, and this is the kind of. So now we've got a big data set of saying that things that aren't antibiotics and things that we can say are antibiotics. I think. Um,
Yes. Yeah. Absolutely. I mean, from this crude assay, right?
yeah.
It's a really like the definition isn't fancy. The definition is a very simple assay, right? But it grounds the learning in something that right that we didn't know before, right? This is the utility of the learning algorithm, right? Because from this small set, we're hoping that the computer will find relationships of like we'll be able to identify what is important and what isn't important in a way that were not able to.
Yeah. So now we've got these two like chemicals, and we can now tell the AI that one's an antibiotic, one isn't an antibiotic. So, and
Mm-hmm.
then
It's going to play around with those knobs,
yeah,
right, to make sure that whatever weights that it's choosing in that fingerprint algorithm, right, in the directed neural network, is able to distinguish between these two classes in the set of data that we gave it, right?
yeah, that's right. Uh, and of course, it might pick up different things depending on how many what you run it on, and you want to make sure it gets the biases least. So what they did was had an ensemble of different AIs where they each they each were trained on different parts of the data set, so they'd pick up like different things, and because they
Mm-hmm.
pick up on different things, you can test them and almost average out, so they won't. So if they are biased, it would average out, and
Yeah,
so
I think that is that's definitely the idea behind ensembling. because again, like we don't really know what weights the computer is going to choose, right? I think actually, wait, let's see. I want to see in the when they ensemble it, did they really subset the molecules to
Yes,
the
because
ensemble?
they said they deferred it. Um, so they have a quite a big section on ensembling, saying like, so apparently they used ensemble.
Different random split of the data is what they
Yeah.
said. Hmm.
Uh,
Yeah, so it's like another way of trying to mitigate bias,
mm,
right? That like we don't know where the bias is. I think that's maybe an important part of like the understanding why ensembling works.
mhm,
Like why should the average of random subsets be better than just running it once over the entire data set?
mm,
And I think the important thing there is that we don't know where bias is in the data set,
mm,
right? Like the whole point of running machine learning is we don't know what features to look at. And if we don't know what features to look at, we don't know where the bias could exist. It could be anywhere. And then the way to get rid of that is adding more random noise by randomly subsetting from the training set, right? We would get random biases in each one, and then the average of it would be hopefully less biased than if we just gave the whole set by itself.
yeah.
And actually, again, this is empirically determined. It's known that this technique works because when you do these tests on training sets of data, right, it gets better results, right? Ensembling gives better results, and I think that that is the strangest thing for me when I think about AI and like a conception of what AI is, right, is that there's a lot of stuff that's done empirically,
Mm,
right? That there's no first principle, like they it sort of becomes the like these practices have become like the language of making good
mm,
algorithms, right? Like it's oh yeah, it works. This is how it's done. You randomly subset, you introduce this bias. I think there are sometimes mathematical underpinnings for the reasons to go down these paths, but it's not always the case, right? Some of it is just by testing it against these training sets and these problem sets, right? Like how was this algorithm found at the beginning? Because they had these sets of data that were um, that are basically for benchmarking algorithms, right? Sets of things that have like right there. People may have heard of Kaggle, right? Like these big competitions where it's just like, can you categorize these images as cancer or non-cancer, right? These exist too for the chemical field,
mm,
right? Can you categorize these things as soluble or not soluble? Can you categorize these things as X or Y? And this algorithm, this directed message passing neural net, right? Like it has performed well in these cases, and it's already known that ensembling them works. So again, this part is not new,
mm,
right? The paper is not contributing, right? A new way of doing this analysis. This already exists. You can get it on GitHub. They actually make it accessible
mm.
for people, right? Link it in the paper. The contribution of this paper is using the tool, right? Training it in a specific way, and then following up on those leads.
Right. So that brings us to Figure 2b, which is an AUC curve, which essentially tells us how reliable the test is. So the true positive rate versus the false positive rate. Um,
Mm-hmm.
so this is where they take it from the test set. They've got a small subset of it that's the validation set, and then there's another that's like a, a further test set. That so
Actually, I think the way that these are made is they actually the algorithm doesn't have to give a binary. So
right.
like antibiotic not antibiotic is a
Mm,
binary, right? That's how the training set was. That's how the algorithm is
mm,
coded, right? So like in the training set, you have all the different molecules, and you have one or zero if it's an antibiotic or it's not an antibiotic. But when the model runs on a classification task, right? So this is a classification task now, it outputs not just a one or zero, it'll output any number between one or
mm,
zero for the confidence of what it thinks it is. And so when you rank, so then they just rank them. Everything that the algorithm spits out, you rank it and you plot that on a graph. And the idea is that you want a lot of confidence. You want the algorithm to pick up things that it thinks is working. And that's what you get with this with this AUC graph.
yeah.
So the algorithm is working is basically what you get from this. It is doing some sort of classification.
Yeah, and that I think brings us neatly to C, which actually shows us like the ranked molecules and which ones the algorithm think are very good fits for antibiotics and which ones it doesn't. Um,
Hmm. Hmm.
so
Yeah, yeah. See that 1 to 0, right? Like there's a bunch. It can fall in this range.
yes, so they took the top 99%, top 99 molecules that they thought could be antibiotic, and then they tested them. Um, and
Oh wait, I guess I want to say, going back to figure C, is that they also, you can, this is the output of a machine learning
mm,
algorithm at the end of the day, right? At the end of the day, this is what all machine algorithms make, and you can look at that in an Excel table. That's what all those tables are, right? They basically did this process with their algorithm and other algorithms, and they put it all in tables, so you can see how I guess the algorithms change the rankings. That's like the whole point. They never speak to it directly in the paper, right? They speak to it of some things once they have their hit, but yeah, I just want to throw that out there. That like this is the output of all of that machine learning is this.
yeah, and so now they kind of took the recommendations and then they tested them to see how well they stood up. And there's
To the real world.
yeah, and actually it seems like they it was actually seems to be quite good because they got about 51 of them showed some growth inhibition based on like a cutoff of a zero point 0.2 for OD600. So uh, that and so yeah,
Yeah, I'm not sure how that stacks up to other things, right? They don't actually do the right? Like, I guess it's really laborious to do this for all the other ones. And again, this paper isn't trying to prove. This paper at the end of the day isn't really proving that they like their algorithm the best, right?
mm,
Like, there's already all this previous literature to say that this algorithm is good at doing this task, right? But you could imagine that, like, if you wanted to know if this algorithm is really good at this task, you would want them to test. You would want them to find this true positive rate, this empirically determined true positive rate for every different algorithm, right? And so, like, yeah. Anyways, that's just like an interesting thing where you can see that the focus of this paper is not really proving that the algorithm is the best algorithm. It's just saying that like this is a valid approach, right? It's more of a proof of a concept than like giving us like an understanding of what the best choice would be.
yeah. I mean, this I think that's highlighted a little bit by Figure 2E because that graph kind of shows the rank of predicted molecules. So kind of towards the left is where what the software thinks would be good, and then we actually see that uh, that actually there because I mean in an ideal world it would basically be if the prediction was perfect it would just be one line that go from red to
Yes.
blue, but the fact that it's two
Yes.
lines shows that there is some area like some wiggle room here. Um,
Yeah. Yeah, like these two graphs, if you could make these two graphs for every algorithm, that would be the data that told you this is the best algorithm to use. But in the way that they've presented to us, they just show us like it's a pretty good algorithm, right? But we don't really have anything to compare it to.
right.
And again, that's not the focus of the
Mm.
paper, right? Like they're just saying this works,
Yeah.
and so it is. Yeah. They, I think they do say that they're really, they're happy that they see that slight upward slope
Mm.
in the graphs, right? In E, because that slight upward slope means that the strength of the prediction does matter at some level, right? That like the stronger the prediction, the stronger the inhibition. So like there's something there.
Yeah.
The network is giving us some information. It just doesn't always work.
And also, if you like look at the density of the dots, you notice that they do like if you just look at the density instead of like just the line itself, that actually does bear out the algorithm's choices a bit better. But
Absolutely.
but,
Yeah.
but because
Yep.
we're humans, we don't weight. It's hard to weight that the way we used to look at the line rather than how it's actually weighted based on how many hits and non-hits it gets.
Yeah, 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 for sure. Like I think that's a good, and they tried to emphasize that with the bars, the color
Yeah,
bars, right? They're like, oh, you might not be able to see this, but look, like also the lines are getting less dense as it goes on and denser in the negatives. So like it can find these
mm.
extremes. It can find the extremes.
And also I'd argue that Figure 2F is almost even more important because that picks out candidates from the least likely pool.
Hmm.
Because I think
Hmm.
because one of the things that I'd automatically say as a criticism would be okay, well, how do you know that if you don't just put like kind of pick out any random number of molecules, you'll find antibiotics? We have to like have a negative to confirm the positive, and this is where they look at the things the algorithm definitely thinks aren't going to be good antibiotic candidates and test them out. And there is definitely less good antibiotics candidates in here by the looks of it.
Yes, but I guess also I think this is also important for people's understanding of machine learning algorithms, right? Is that like there still are things in here that have biological relevance, right? That the computer doesn't pay attention to. And one could ask, why? Right? And the answer would be because the training set, right? Like, I guess like this is where it gets really complicated, right? Because like, why is because the computer doesn't think they're important. That's because we gave the computer this type of training, because we gave it this information. This is the space in which improvements could be made as well, right? Like understanding what made these things go out, could be something, could unlock other things. Again, this is if you're like a computer science researcher, maybe, right? And trying to make the best antibiotic discovering algorithm, right? Like you'd be stuck in just this part, right? Like you'd be iterating over these pieces of data and back to the math drawing board, like playing up with the algorithm. But we're microbiologists and we're pushing forward because we want to know like what about these? Like, did it find novel antibiotics? Right? Like, are these antibiotics going to be useful? So let's keep going.
Yeah, so now we're going to go. We got like a network map showing the relatedness of all the molecular structures and the data sets they looked at, and
that's like we've seen these types of plots before. It's dimensional reduction, right? They use TSNE, but we've seen UMAP before, right? It clusters things that are similar.
Yeah, and what they find was that they did find an antibiotic from this. So they screened out a bunch of things that they actually screened for things that are being tested in clinical trials.
Mm-hmm.
And so they found one of their positive hits was this C-gen N-terminal kinase inhibitor called SU327. Anyway, they call it halicin, and I'm going to stick with that.
Yeah, we don't have to call it. I think it also had like it had a designation like SD something something.
SU3327 is its designation.
There you go.
Yeah. I've got my notes in front of me. So it's a preclinical nitrophiazole under investigation as a treatment for diabetes. So this potential diabetes treatment turned out to be quite a good antibiotic, but based on what the computer predicted.
Yeah, just strange, right? Like I guess it just shows you. You know what I also love about machine learning is it also shows us like the bias inside of our own investigations as humans, right? Like we can go like we can investigate things. We only see in science what we ask. And so we're asking questions, and they're good questions because they're based on prior knowledge. But if you took a step back and you took this like crazy unbiased eye, which is the machine learning algorithm, right? It can find relationships and useful things in the mix that we just ignored, right? Like why would anyone try this diabetes drug, right? In this setting. I mean, I guess they say that it's similar to metronidazole, which is a
nitrophiazole. I think kind of it's got a similar kind of structural thing. And the thing is, like, if this failed in its diabetic trials, it'd be disappear and just like end up in the drawer. And so and that's kind of part of the reason why drug repurposing is so interesting and important, because sometimes you discover a molecule and think it'll do one thing and it doesn't. And frankly, other researchers come back and find that it does something else, and that's kind of what's happened here.
Okay, let's push forward then. They just make sure that this is working, right? Yep. That they run it through their OD, their inhibition study again, and they find the MIC.
Yeah, so
which is about 2.
Yeah, about 2 micrograms per mil, which is actually pretty good.
Absolutely, and you can imagine that once, like, so this is the beginning of drug follow-up, right? So like this is basically the machine learning algorithm has taken us to this point, right? Was able to get us to this point. This is where regular hit screening would also get us, right? It would bring us here, right? Where we're like, okay, we found them some things, and now we're going to follow up on them a little bit more. And after that, they can do all sorts of things to drugs. You can try to improve its solubility. You can try to improve its features. You can design other drugs that look like it. So like this is we're kind of seeing where machine learning is catching us up into the pipeline.
Yeah, this is where the paper turns into a straight microbiology paper for the most part, where they found this new antibiotic, which is a really important thing, because again, we don't find many new antibiotics these days. So finding one with which is kind of dissimilar to other structures, is very important. Um,
Yeah, really. We want to learn about it,
yeah,
right? Like now that they fished out this molecule, like we want to say, how does it work? Like, is it really new? Can we say that definitively? And that's yeah, we're gonna see a lot of that coming up
yeah.
next.
So coming up next, we've got it growing in. Uh, so this is figure S3A, and okay, so what's happening here?
Bactericidal. So
Yeah,
like they were doing MIC, right? Like this is where you said lysis at the beginning. They never touched lysis,
right.
right? Like they were only looking at inhibition of growth. But now there's a difference, right? You can stop something from growing, but then after it gets diluted away, it grows
Hmm.
again. A nice feature of an antibiotic would be that it kills the
Hmm.
bacteria. And so the way they do that is they grow up bacteria and then they incubate it, so they grow it to like a density and then they incubate it with some of this compound and they see what you were saying before, the popping or the lysis would then make a decrease in how many bacteria grow again, I guess.
Yeah,
So it's not even OD, right? They actually plate them again to say like, are they alive anymore?
yeah,
um,
and
and it works. It's bactericidal based on this
yeah,
task,
and they
right?
test out different concentrations of it, and they find that it does change the dosage a little bit, but the important thing is it's bactericidal. Um,
Yep.
and next one they're looking at.
When you say like it changes the concentration, like it does it in a way that's favorable,
Yeah,
right? It sort of says you decrease the concentration of bacteria, you get more killing, meaning that you like more of this compound does the killing. It's a
yeah,
logical relationship. It's a
that makes
logical
perfect
relationship.
sense. And they do it in rich media, and they also do it in depleted media. So rich media is where the bacteria get everything they want to eat, and they can grow and be very happy. So it's kind of like inside the body, but also kind of the inside body is a complicated medium. So they also use PBS, which is basically a nothing medium. So trying to look at kind of the highs and the lows. So where a bacteria would really exist is kind of in between these two states, I think. Um,
Yes. Absolutely, and it's already. I think the other thing is that it's known in litter. It's right. We know from studying antibiotics before that when things don't metabolize, when they're not actively in metabolism, sometimes you can't actually kill things with an antibiotic in that state, right? And I think a really important thing. I actually will go to the next slide, right? Because this is like we know ampicillin. It's a molecule that inhibits an enzyme in cell wall synthesis, right? And when you inhibit that enzyme, so the bacteria is growing, and it's like, oh, we can't make cell wall anymore, but it's still doing all of those steps that it would do in preparation for division, and that ends up killing the bacteria, right? So ampicillin is bactericidal, but it requires the bacteria to be doing
mm,
stuff. Because if the bacteria is not doing anything, if it's not growing actively, then the cell wall inhibition doesn't do anything because it doesn't need to make cell wall. That's not an important molecule. And so in this one, the top wall, right, the top, oh, this is just the example of
yeah,
ampicillin, right? Ampicillin doesn't work in the nutrient deplete
right.
conditions, but it does work in the nutrient replete conditions.
Yeah, completely. So that's a good point of comparison that we can have for Halison. Um,
Mhm.
so figure 3C is almost like comparing a combination of Halison and ampicillin. So yeah,
It's like, and I think important to look at the time, right? That
mm,
it's sort of like, oh, if they have all these bacteria and they put some ampicillin in, after a certain period of time, there are some cells that just don't die from it.
yeah,
And if you add Halus into it, then they do die. So I don't know. They say persister here, but like I know persister is like a very strict.
mm,
Like there's a lot of stuff that goes into defining a cell as a persister. This is one persister
mm,
phenotype, but it's not all the persister phenotypes. I would say.
yeah. So persister is almost like before it evolves antibiotic resistance, where like it's kind of withstanding that big shock of the first antibiotic, kind of.
Hmm.
Yeah, yeah. But yeah, over a certain set of hours, I feel like, I mean, because it really depends on, because I know Ecoli has a doubling time of like 20 minutes in certain media at its right temperature. Um, so that so for an
Because like what it could be doing is it could be killing ampicillin resistant bacteria, right? That's a possibility.
yeah,
Yeah. Yeah.
but the thing is, like, I guess when they add the halicin, they literally derail that, right?
Yeah, Hansen works on those things. I guess I just had a problem with this figure because, like, I could tell that they were trying to speak to this body of literature about persister cells, but I also think that they didn't do the experiments to really speak to that body.
Yeah.
They gave us a crumb, right, where I'm like, oh yeah, like this could deal with persister cells, and maybe we'll get to a paper about persister cells in a later journal club, but it is a big body of literature that ways of describing those cells. I'm not sure if I think that this is like a really good description of that.
Yeah, I think that's fair. Next one, they look a bunch of different. So this is figure 3D, where they
Mhm.
look at E. coli strains harboring a bunch of antibiotic resistance determinants. So
Mhm.
this is where they test whether any of these are actually able to break down halicin. Can any of these antibiotic resistance protect these bacteria against halicin?
Yeah, and it doesn't really seem like it. So they have the controls right on in K where you can see like those resistant bacteria, they really grow a lot more than the wild type. And you can see with halicin, it's still all around 2. In some cases, it does better. I guess that must be noise or something like that, right? But it even seems like the MIC is better in some of these circumstances, probably because those bacteria can't grow really well anyways. There's some other things that go into that, but importantly, none of it. There's no jump upwards, right, above 2. So from this data, they can feel moderately confident that the mechanism of action of halicin is not one that we know
Right.
before. Mhm.
And so they next one they moved into tuberculosis, into Mycobacterium tuberculosis, to see. And they again like they do their kind of MIC and they find that okay, it actually does pretty well against tuberculosis as well.
Wait, this is No, this is not
Oh, sorry, no, you're
tuberculosis.
right. I'm sorry, I got completely confused there. This is a completely different one. Uh,
This is just, I guess, I don't know anything. I don't know too much about this antibiotic, but apparently you don't give resistance to this antibiotic by giving them a plasmid. You do it by knocking something out, or you put something in the chromosome. And so, like, this is just showing it's another antibiotic that it doesn't work on.
oh, right, yeah. Okay.
Yeah.
Yeah, sorry. Forget what I said about tuberculosis. This is something completely different. So yeah.
Yes. Yeah.
So
This is another extension of Figure 3D is this L and M. One more antibiotic that halicin does work on that usually one more antibiotic resistance mechanism that halicin doesn't care about.
yeah. So and this is oh yeah. No, this is the tuberculosis one.
Then we go to
Yeah.
tuberculosis. Yeah.
So yeah, this is where they test on tuberculosis and find that okay. And they also test it on a whole bunch of different other resistant. So they looked at tuberculosis. They also looked at sorry, Acinetobacter. Yep. Pseudomonas
Pseudomonas aeruginosa and carbapenem-resistant Enterobacteriaceae.
Yeah.
And these are all like offenders, I guess you could say, of antibiotic resistance, right? Like tuberculosis, this strain that they're using is like multi-drug resistant. They have this whole panel of these different organisms, right? Every bar here is another organism that is resistant to something. And so across all these panels, it does seem to have inhibitor concentrations, but you can see that it's not really at 2 anymore for some of these. So like especially Pseudomonas aeruginosa, that MIC now is at like almost 100.
Yeah.
It's probably in the 50s or something like that. So we're getting into the range. We're taxing it, right? Like it's not going to be perfect. They throw out the hypothesis that something about the capsule of Pseudomonas aeruginosa might be the reason why halicin doesn't work on it. Who knows at this point, right? Like you just don't know. But in general, you can call it a broad spectrum antibiotic maybe from this point.
Yeah.
It's working on different bacteria. Is Enterobacteriaceae, are they gram positives?
Uh,
Are they gram positives? I'm not sure.
I mean,
They don't.
sorry, I do like kind of wish they had more positive controls on these graphs so I could like keep things in
Yeah,
context. Um,
yeah, absolutely. So it's a good point. Like, I do think that they're kind of running away. They're really excited about what they found. They're showing us a lot of different things. And as I said before, like they're giving us breadcrumbs to these phenotypes, but I don't think they're like definitively nailing down this is the phenotype, right? So yeah, broad spectrum, but like, what does that really mean
yeah.
given the data that they've shown?
Uh, so
Okay, we're gonna they're gonna plow
Yeah,
onwards.
plow on.
Yeah,
So figure 4A.
they want to
Uh,
the mechanism of action.
right. Yeah. So how? So they figured out that it works. How does it work? Um,
Hmm. Hmm.
yeah, so first step is trying to evolve halicin resistant mutants. However, they're
Hmm.
unable to isolate spontaneous suppressor mutants after 30 days of. So usually you just like try to incubate your bacteria in with your antibiotic for a fair amount of time, and then it'll eventually evolve a way to get around
Yeah,
it. But apparently they
yeah.
couldn't do that, which is kind of uh, show offs.
Humble brag. Right? Yeah, this is a humble brag, and
Yeah.
I also think it's not. Again, I don't like when I appreciate that they've done this and they've shown it to us, but again, it's not
Hmm.
definitive, right? Like if you want to prove that something doesn't make resistance, you actually do like there's different experimental designs to go down that path,
Yeah.
right? There's different gold standard experimental designs. In this one, they're comparing it against the evolution of resistance in Cipro,
Yeah.
right, on media and in liquid. But yeah, I don't know. There's more to do to say that like you can't evolve resistance, right? More so, so like I like that they did this because it is interesting to know that maybe it's more difficult to evolve resistance here, and it ultimately is serving in the paper just the logic that they can't do it this way. It's more difficult to go down this route to find, so they're going to use a more transcriptome focused approach instead.
Yeah, so they take out all the RNA and sequence it to see what's different when we add this antibiotic. And I think they
Yeah.
found that genes enriched, so they split them into different clusters. So some genes were related to locomotion and how that works. Others were related to iron homeostasis. Um,
Yeah, and so this is I kind of like this is kind of cool. I like it if you can't find the chromosomal changes, right? If you can't find mutations occurring in the chromosome, then maybe you can just find like upregulation and downregulation as a way of getting at mechanism. It's kind of cool. I like that interplay between like because in my mind transcriptomics is like a very new genetics
hmm,
like view, right? We weren't able to capture this information before, right? People if we were back in the olden days, we would basically say, let's just continue trying to find suppressors. They didn't try hard enough to find suppressor mutants. And you just keep doing it until you found it, right? And I think they would have eventually found it, but this is just faster now. I
yeah,
think this paper is a really funny study in like how technology has changed a lot in the way that we approach these questions. Like now when you can't figure something out, you're just like, well, let's just do a different method. Let's get all the data, let's get data in a different way. And this is something that, yeah, I just love seeing this in action because it's like a cool evolution of science milestone.
yeah. So based on this data, so they know that there's something happening in locomotion. And locomotion is related to kind of the protomotive force. Like a lot of things like flagella are powered by that protein gradient. So this comes back to that foreshadowing of that protomotive force, which is kind of governed by two things. So firstly, there's the electric charge. So you got your hydrogen ions on one end of your membrane and lack of them, and then that has like an electric charge. It also has like osmosis, where simply just having an ion of a certain thing being having a difference between them, that also mean has a proportion. So
Yeah. So I think a nice way to think about the electric potential is that's just charge, irrespective of knowing like a specific ion. So like things like sodium ions have
hmm,
charge, right? Potassium ions have charge. If you selectively export those ions across in a different direction, you can build up charge on the membrane. And then I like think of pH as very specifically hydrogen
hmm.
ions. Right? Because like that's very specific. It's like that's only talking about hydrogen ions and the concentration gradient for those particular things. Um, and yeah, it's kind of like a battery. It's a capacitor, actually.
Hmm.
If anyone's into electronics, like, right, this is like a capacitor thing, right? Like the membrane acts as a capacitor, it builds up charge, and then you can have little leaks in the dielectric or the membrane in this case, and then you can use the charge that passes through as energy.
Right. So how do they test this? Well, uh, the first.
Sorry, one more thing I want to say about PMF. So cells have many ways of regulating PMF, right? Because it's really important for them to
Hmm.
survive. So usually, if you do a change in one, right, you'll get a change in the other,
Right.
right? Because the cell, in trying to survive, will start like upregulating different mechanisms to try to like, or different transporters will turn on, right? So like, just keep that in mind as well. They're related.
Right, for sure. Um,
Mhm.
so yeah, so since I mean, so what are they measuring here?
So since they think it could be proton, since they think it's PMF maybe, right? They one of the hypotheses you could say is that then changing the pH, right? Because that's changing the concentration gradient of hydrogen from the inside and the outs from the outside at least, right? Changing that would maybe reduce the MIC activity.
Right, and they kind of so as they increase the pH, which they would therefore kind of decrease the amount of hydrogen ions in the solution, halicin becomes less effective. Um,
Yeah, and that's consistent, right? Because if halicin is like breaking up the hydrogen ion gradient, right, it means that it's equalizing it. It's probably letting hydrogen ions into the cell, which would then make the two concentrations equal. So then, oh well, let's just compensate by that by removing some hydrogen ions from the exterior, right? And then maybe that means that halicin can't do that as well, like break down that
right.
barrier. That's what they're seeing here. So that's how this ties into their hypothesis. But it's like a scrap. But they do a good job about following up on this particular hypothesis.
Yeah, I mean they tested both Staph aureus and E. coli, I believe. So
In figure S3E, they do it a different way where they just add in sodium bicarbonate to lower the pH, like as it's growing, and so that's a way. It's basically the same point that the pH is lower.
and figure 4D, they add a potentiometric fluorophore that basically accumulates in cytoplasm when there's a charge differential across membrane, and when that charge differential is disrupted, the
Membrane.
membrane.
I think it's membrane. Yeah.
Yeah, so a charge differential across membrane, that's what we've talked about before, and when that's disrupted. So if there's a hole in the membrane, or if that charge differential is equalized, that fluorophore can go into into the media around it, I guess.
Yeah, yeah, and it self quenches there. So they compare it against. So they have a positive control, right? In each case, the valinomycin for Staph aureus or the polymyxin for E. coli. Those are two things that just like break up membranes, and so you get the release of the dye and that increase in fluorescence. And then they have halicin, which when they add it, the fluorescence goes down, and so going down would mean that more of the dye is going into the membrane, right? More of the dye going into the membrane is actually that delta psi, is the electric potential
Right.
changing, right? So cells are like, what they're seeing here is that like when they add halicin, the cells start trying to do something to their charge balance, right? They're desperately trying to hold on to like the charge in some way, and because uh, they they hypothesize that that's through the pH mechanism, right? Like that's why, or that's sort of the reason why seeing this signal, right, from the electric potential side is giving them more confidence that it's the pH side of things. So yeah, they're not changing pH now. They're just seeing like that it's working on the converse part of that equation on the other half of it.
Right, right. Um, yeah.
So I good, right? So like this is where it's like I feel like this paper had like some breadcrumbs where they were saying like persister cells and like broad spectrum, but like this one, I feel like this is the gold
Mm.
standard of saying that something is acting on delta pH or on the PMF. So I like this. This is to me like the most important graph of like how the mechanism of action for the mechanism of action
Yeah, I
story.
mean, I think it's really good. I mean, just looking at the timing of it, because it's almost an instantaneous effect. They're measuring in like seconds essentially,
Mm-hmm, mm-hmm,
which
mm-hmm.
is like kind of so these are the immediate reactions of these cells, which I found very interesting. Uh,
Yeah,
yeah,
and the thing is that it stable. I guess never mind. This doesn't tell you anything about like the if the cells are successful in dealing with this charge, right? Because we're just measuring the fluorescence of the dye,
well,
so we just know that. dye is getting absorbed into the membrane.
Well, yeah, well, this like reflects the charge aspect of it because the pH reflects the chemical aspect. So, as you mentioned before, cells have ways of altering their charge. So that charge is returning back to normal. But that doesn't
Mhm.
necessarily mean that the hydrogen ions are equalized. That could mean that there's other ions inside that are doing that work.
Yeah, for sure. Yeah. And then those actions could be so like this is the next level deeper that we don't get, but like how does the disruption of PMF by halicin kill the cells? Right? You could ask that question as the next step.
Yeah.
Um,
So
okay. Oh, they're going to give us some more like more correlates, right? To this being a PMF or a delta pH issue. And that's because they look at the synergistic or antagonistic effects on two different antibiotics. And we know previously that tetracycline is something that is dependent on delta pH to get in cells. So because it has an antagonistic effect on the killing here, then we say, oh, it must be disrupting the delta pH. And then we see this like synergistic effect with kanamycin. And because kanamycin uses the electrostatic to get in, then this is that compensation that the cell is doing, right? When halicin goes in, it compensates by like pumping stuff in or out, and kanamycin gets in more there and has more of a killing phenotype.
yeah, which kind of fits in with what we've seen before because we know that the pH is changing, but the from what we saw immediately that the cells adjusted the electrostatic part of it, so kanmycin could still get in. So that's quite interesting. Another interesting part is what they looked at with iron. Because when they increased the amount of iron available, that that made it more effective at killing. Um,
Yeah, and they think that that's similar to a mechanism of action for daptonomycin. And so then they ran this little test where they're like, oh, well, what if we, right? Like they didn't test like if daptonomycin resistance, right,
yeah,
is going to have an effect. And it kind of doesn't.
yeah.
Kind of
But I
doesn't.
mean, it is interesting because like one could speculate that the ions could be the thing that is changing in order to keep equalize it. But I don't think they've gone as far as to say that. I think the most they say here is that it could be like almost like a cofactor that helps the halicin killing.
Yeah, they think that's what's happening not inside, right? Because like a deletion on the chromosome, I actually didn't read into what the actual mechanism of action is of the delta DSP1
Mm,
resistance. Like you could say like, oh, but that's something that's affecting the intracellular environment. But when they supplemented the iron in, that was in the extracellular environment, right? So like, okay, it kind of works like daptonomycin, but maybe just like it's sensitive to a different thing. This is a great figure for the next paper in the mechanism study, right? It would be to like grab some of these results and go down this route. They definitely pursued this more than they did again persisters or evolution of resistance or something like that.
yeah, I think that they're more interested in figuring out the how rather than like the because I think with persisters we're looking for okay, is there going to be resistance? But I think they're more interested in finding out how this new antibiotic works.
Yes, absolutely.
So
Um, and again, because I'm always struggling to say like, what is the paper teaching us? What is novel about the paper, right? And like, this is part of their novelty, right? Is that like using this method, they found something that really we haven't really seen before, right? All these tests kind of fail to describe what we've seen, and that's exciting, right? The finding of novelty, because as we said before, people have hit a bit of a brick wall in finding new antibiotics. So yeah, it's very exciting to see this data.
yeah, next they look.
So they're gonna go down the path of showing us some clinical relevance or preclinical relevance of it. And before they do that, they're gonna identify the bacteria that they're gonna use in their model, which is this multi-drug resistant Acinetobacter baumannii. And we see some of the same in vitro tests that we saw before. The MIC curve, it works against it, and the bactericidal tests. Again, it's bactericidal.
Yep, and then they take it into a mouse model. So a mouse wound
Hmm.
model that is using this thing called tape stripping, which I shall demonstrate for you now. If you can, so watch very closely. Take a bit of sellotape, and I just take it off, and this like repeatedly put it off in order to create a wound model. They use a much stronger sellotape, but effectively that is how the model works.
Yes. Mice are also neutropenic, so
Yep.
like they also don't really have a good immune system. And so once you've done this strong tape stripping and dripping of bacteria onto the wound, these mice cannot control the
Yeah,
bacterial
so yeah,
growth.
they made these nice neutropenic. So I think for these models, they have to make it a bit able, because I guess in normal situations, mice have a lot more neutrophils than humans, and that means that humans tend
Mm.
to be more susceptible to things like Acinetobacter. So in order to
Mhm.
get you can tell this is a part I know more about because I've been silent for quite a lot of this, um, but yeah, so they drained it and then they created this model that's been tested before, and they tested whether their antibiotic worked, and it, and so they kind of did a topical like kind of it's effectively like they've bruised the skin a fair bit and added the antibiotic at certain time points to see how it reduces this bacteria.
A lot. They put a lot of antibiotic
Oh
on it.
yeah.
I feel like it's hard to say that this model, like every 4 hours for 24 hours. I mean, in a hospital setting, that's definitely a very
Mm,
realistic type of application schedule, right?
right.
Like I could see this being like, I know burn is like a big thing where like you get resistant infections appear. Um, uh, what is it? Diabetic
Yep.
ulcer, right? Is a wound model where this is really important. And so in both of those things, I agree with the way that they've applied this. It's like hospitalized settings, right? And again, that's actually where there's a lot of fear of antibiotic resistant bacteria, because in those settings is where we have a lot of those strains circulating, and we also have people that have various levels of immune compromised or like otherwise compromised functions. And so, yeah,
Yeah.
it works in the preclinical model of that scenario.
Yeah. Um, so next they looked at a model of C. diff. So C. diff is a really big one for because C. diff can form spores and effectively have this state where it's not very susceptible to many antibiotics. Only metronidazole is the only one that really takes it down. But usually it occurs after you've given a patient lots of antibiotics and cleared out their normal gut bacteria, and then C. diff can outcompete them and cause a disease called pseudomembranous colitis. Um,
Yeah, basically it grows so much that it's making its own layers, sheets of C. difficile in your intestines.
yes, it is unpleasant. So, yeah, they found that the C. difficile is actually quite sensitive to halocidin, which is quite important. So they did a model of pseudomembranous colitis where they gave these mice a lot of antibiotic and then waited for that to come out of their system, and then they infected them with C. diff. And C. diff proliferates when it doesn't have. If there are other bacteria there, it doesn't do so well. But if there's nothing, if you give it like plain and empty like intestine, it'll just do great. And then they tested their antibiotic on this model.
Yep. And they give it a nice they have a positive control, or they have like a comparator in this. They have metronidazole, which is the sort of the standard of treatment now. Although if people are following the news, you also know that like fecal transplant is like the new exciting technology for dealing with C. difficile.
Yeah, eat shit, bro. Sorry. No, that's a scientific term. It actually counts because it's scientific. It's
And in this model, it works. Like by day 120, they have none of their animals have detectable C. diff anymore if they're treated with
I mean, I
Hallicin.
think it should be noted that 3 mice in this group initially didn't get C. diff infection, so
Mm.
they had to take that into account a little bit. So they are working with a small group size, and there may have been an interfering effect that made those mice less susceptible in the first place. So that's something that we need to take into account. But apart from that, in those 3 mice, it did look like it was doing quite well. Um,
Yeah. I guess like again, like this is not like these. This is again. They're back to the breadcrumbs
mm.
in this in this paper, where it's like these are really interesting results, right, to get, but they're also not the full like characterization of these models, right, and this
Oh,
particular
yeah.
drug.
I mean, a lesser research lab would have been able to like salami slices into like 10 different papers and piecemeal pass them out. But they're trying to like give a whole picture to try and almost uh, get like the hype up, get a bigger picture about this. I
Yeah,
think.
they're very like this proof of concept is a really big like line that runs through this paper, right? They do want to do good science and show us like hints of mechanism and stuff like that, but they also want to say like people should be doing this, right? More people need to be doing studies like this, right? And following up even more because there's real potential here, right? A big part of this paper is the potential.
Yes, exactly. Because yeah. So I think moving on to the next paper, where next figure. Oh, now this is an interesting one where because now we're going back to the
Yes.
AI.
Backpedal to the AI, right? And really, like in some ways, maybe people were feeling a little bit cheated because when they did their initial screen, they only used like this drug repurposing hub data, right, which had 2,000 things. But if we're really going to make a dent in like the pipeline of discovery, right, we're going to have to go orders of magnitude larger than a few thousand things in a library. And so here they have anti-tuberculosis library that has 10,000, almost 10,000 different compounds in it.
And the interesting thing about that is they didn't find many good candidates in that one, which I
but they also knew that this library is structurally divergent. And so again, going back to the sort of the mess of trying to explain the algorithm, but it's already sort of de novo, you know why this doesn't work, right? Because if the training set doesn't have the complexity, right, doesn't capture the diversity of the classification problem, it's not going to work, right? Like you're not going to have things that classify well. And if you go to the next figure 5, S5, B, and C, when they take the top 200 hits and the bottom 100, none of them have antibiotic activity, which isn't surprising because the model wasn't very confident anyways of the things.
I think the fact that it isn't surprising says how much we're convinced of this model at this point, because, I mean, for me, if I was thinking this model doesn't work, then it would be surprising, because I'd imagine that there'd be maybe one or two things that are random, but this one actually almost shows that the model might have some sense, that it's, because you'd like to think that in this library there may be one or two things that might make Ecoli like think twice before growing, but
yeah.
But the fact that like the model can show that, okay, the model says it's not confident, and then you can then demonstrate it, that I think that's quite an interesting thing. Like
maybe, but I'll counter with this, right? Because so they did the top 99 and the bottom 63, right? In the drug repurposing hub, right? And that percentage of the library in total is a bit higher than this percentage, I think. Like actually here they should have gone up by one order of magnitude. If the library is like 2,000 or whatever, they should have tested more, right? And then they also used hits from the library, from the predictions, right? So the predictions are picked out of this list non-randomly, right? If they picked 300 random things and then they showed that there were no hits, then I would say, okay, that speaks to this library itself.
Right.
But actually, the algorithm could just be very confused by this. It could be picking out things that just don't match up at all. This library still could have stuff in it that's useful. We just don't know, right? And here's the big challenge, right? This is the challenge that we're trying to solve the whole time, right? Is how do you go through a huge amount of molecules and find something that works, right? And how do you do that in an unbiased way? This is a limitation. I think actually, to me, this is a good point that they're making about the model by showing us this data. They're showing us a limitation of the model, right? That the model can't be used to find things like this. I don't buy it that it helps us say that the model is good, right? Yeah. I think it's more just saying that this is a blind spot, and there's no way to address it right now.
Right. Yeah. Because for a moment I was thinking, like, we're the most nightmarish reviews ever. Oh yeah, can you do 10 times more experiments to support this? But
yeah, I don't think that's it.
Yeah.
Like this data is important, and it shows us that it just doesn't show us. It's not telling us that our model is good because we know that this data set doesn't have any hits in it, and it didn't find any hits in it, right? We actually still don't know if the data set has any hits. I guess a priori we know because it's anti-tuberculosis medicine. Right, like people think that it's not going to do anything, but again, I don't know. Like that's a lot of things, right? Like a different algorithm could find it,
Yeah,
potentially.
I think you're right. I think there should definitely be a hits in this library. It's just that this algorithm, based on what's been trained on, isn't equipped to find them, and that's
Yeah.
why it's picking up. So yeah, I think. I mean, I think the interesting thing is that kind of random transect idea of just like picking out random and then see how that compares. Just to get.
Because again, we don't know what the algorithm is prioritizing in terms of what features it thinks are important for antibiotic resistance, right? All we know is that those features are tied to the biases of the training set. And we know a priori that this training set is not like the library that we're testing it against. So seeing this negative result, I don't know.
Yeah,
It's right, like it doesn't work in this circumstance, which is an important thing to remember because that is the bias of all machine learning things. Yeah.
yeah. I mean, I think the idea of like doing that random thing to get an idea of the hit density, like seeing like, but then again, doing that, you never know when to stop. So it's.
Yeah. 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 For
But
sure.
anyway, moving on to Figure 6A. So Zinc database, which is much bigger. It's
Mm-hmm.
so one.
Uh, yeah. Now we're getting into that really large. We're saying, oh, here's a even bigger one, and they're going to leverage. I don't know if I said it in a later slide. I don't think I did. So like the important thing is after doing that Wuchi anti-tuberculosis library, they said, okay, well, all of that is good training data, right? So they can pump that in now, and now they've expanded the chemical diversity now of their training set again. And so, okay, now they have another data set to look at, and maybe they're better equipped to look at this data set because they've expanded their chemical diversity by adding all of that negative data, right, into the training set.
Right, yeah. And so the zinc it has 1.5 billion theoretical structures, which is
Which is huge.
huge.
Yeah. And it would take, I guess, probably too long to go through it. So they actually subset this library as the things that they're going to end up testing.
Yeah, and so they filter things down. They're still having to end after all their filtering, churn through about 107 like million molecules, but
Yeah,
um,
huge. And that is those molecules are all indicated. Those tranches of molecules are all indicated by the yellow here. And the way that they filter it was things that they a priori know could be
hmm,
antibiotics, right, and have similarities to their training set. So they're also right. They're also right. They're helping the algorithm along a little to find things in this data set.
yeah, yeah, definitely. And yeah, so I think moving to Figure 6B, which kind of shows out the output of it, where
Mm-hmm. Why did they show it like this? I have so many questions at the end of this paper.
mm,
Why did they show the hits in this way, but not the way that they've shown it previously? Right? Like we just saw before, they use a line to show it, but here they show it like this. It confuses me. I'm very confused why they mix it up for
I
us.
mean, I think it's so we don't get bored. I mean, it's yeah, have different throw different graphs at us.
Because this is essentially the same graph that we've seen before, right? It's prediction value against the molecules, except now I guess they're giving us the quantity of molecules to kind of bin things into like they have a cluster of stuff that is up
It
there.
might be speed because the software output requires it to come out this way rather than the because they were dealing with much more data, so maybe they have to bin it and I don't know.
No, I don't understand. It doesn't make sense. Yeah, maybe they couldn't put the full data set through. That could be it.
Hmm.
They couldn't put the full data set through, but that still doesn't mean that they can't show us the same graph. In my mind, I'm trying to transform this graph into the graphs we've seen before because that's more
Hmm.
comparable, right? That's how we can understand what's actually going on here. And I think what I'm seeing here is that they have lots of hits for one and down. The graph actually, after they've incorporated the Wuchi tuberculosis model into the training set, they get a lot more positive hits, right? You're no longer seeing that like smooth curve where there's only a few things that get positive. They start seeing a chunky curve where it's like lots of things are at high and then it kind of like goes down a little.
Yeah, I see what you mean. Yeah.
Yeah, right. that's the transposition of this graph into the format we've seen before. So the algorithms changed a lot
Mm.
by adding the Ruchi data into it, right? Like it's become a lot crazier, I guess, in its predictions, right? It's saying like it can now predict what it thinks are antibiotics in a larger stretch of things, right? And again, this is an important point I think people need to know, right? Like, and it's kind of hidden in the paper somewhat, right? That like the training set really impacts the types of stuff that comes through, and so much of using machine learning algorithms is in the practice, is in the empirical application of it, right? Because given this data, they're going to have to filter it in a different way than they did before. They can't just take top 100 because top 100, right, isn't capturing the stuff that the model thinks is right. Right. Instead, they actually do a cutoff on the probability score, right? And we can see that in the next. I think we see it in the next one. They cut off the probability score at 0.8, and then they apply another type of score, which they haven't previously needed to do to filter through this data, right? They apply another thing to say we want to find things that are really different from other stuff.
Right, yeah, the Tanimoto score, which is essentially this like is a mathematical way of saying is it similar or not.
Yeah. And from that, they get 23 hits, right? So, like, kind of interesting, right? Like, the output of this model has to be massaged a little bit differently in order to get us to a hit way. And that's based both on how we've trained it, but also probably on the size of the data set.
Yeah,
And I guess for me, because I spent so much time reading about how this algorithm works, like I want to know those things. I want to know their empirical lessons from using this algorithm. And the story that they've shown us isn't those empirical lessons. It's more just like, here's what it can do. But if people are going to work with this in the future, you're going to have to know the empirical lessons. And you know what? As a cynical dude, right, I'll say that those empirical lessons are trade secrets in some ways. Those things are the devil in the details that companies can step in and say, we can use it. We know how to navigate this confusing landscape. And I can't help but look at this paper as a little bit of a marketing thing, right? Where it says, like, there will be companies that come out and say, we know how to massage this algorithm in a way that gives us consistent hits. Right, like this is priming the market, priming investors to learn what the important things to ask companies are, and then giving the companies, right, a roadmap to say, like, we can hit those guideposts and we can make those things for you.
I mean, I say that your cynical reasoning is, I think there might be something there, but I also think that there is a lot of problems in trying to get that those empirical like kind of figuring out what the AIs are actually looking at. I think there's a lot of trouble people go to try and actually figure out what AIs know because
Mm-hmm.
so I think that there's an issue there that it could be.
AI knows. It's not about what the AI knows is the empirical stuff, right? It's what the people know, right? In sort of massaging that data set, right? What if they, for example, right? What if they showed us the prediction score from each tranche individually? What is that all about, right?
Right.
Could they show would that teach us something about what's going on? Again, that's not what this paper is about, right? I think that that's the important point that I want to reiterate is that the paper is not really trying to teach us about this machine learning algorithm as much as it's showing us that the machine learning algorithm can give us useful things, right? But in terms of how to use it, I don't get that. They have the ability to teach us that. They just haven't written that paper yet.
Yeah, I see what you mean because I think another part of this is that this is written almost for microbiologists to try and understand. There's a lot of stuff of trying to break down this machine learning algorithm into very simplistic words. And think about when you're writing for a journal, you have to write towards the audience because it's going to be sent to reviewers who are microbiologists. And so if you throw too much
Yeah.
machine learning at them, they might just go, okay, well, I'm not going to review this. So I feel like
Yeah.
so there are various other aspects. I mean, I'm not saying the cynical aspect is wrong, but I think there are lots of other like kind of other dimensions to look at this through.
Absolutely. No, that's a really good point. Like how could this paper even come to publication, right? If it branched off in too specific of areas, there'd be no one who could actually render a good opinion on it. And so the way that it's written is Cell is a biology paper, right, for other biologists. It's also a very prestigious paper that tells stories, right, that need a lot of figures. And so this is the way the story came through here, is for that reason.
Yeah. So moving on.
Okay, I sidetracked us because I want to talk about. So top 23 hits, they test them in their MIC assay, and they find 8 candidates in there.
Right. Yeah, we've seen these graphs before. We know kind of what they mean, so I'm going to skip over them straight to the 8 candidates. They haven't given very pronounceable names, but
Yeah, they've left them all in their designation in the library state.
Yeah, so we could talk about zinc 0002255434673.
But the takeaway is these are the MICs across. Now they do all the different organisms, selection of organisms, and you can see there are actually 2 that come up that have broad spectrum activity.
Yeah. So again, I've pulled up those graphs. You can see. I'm not going to say the names again, but they do have broad spectrum activity. And going further on, some of them had
Yeah.
So okay.
Oh, go ahead.
Okay, sorry. Okay, I will say the names one more time. Okay, zinc 0001000327116 has structural features found in both quinones and sulfur drugs, but its Tanimoto scores, of course, so it's still quite a very divergent structure. And then there's another one which is kind of a bit more interesting. It's got a very different structure called zinc 00022544673, which is very new and different and potentially low toxicity of humans. So it could be very ripe for exploration in the future.
And they do that preliminary characterization where they see, does it have a mechanism of action that's different than the other resistance mechanisms that we know? Seems like the MICs are very similar.
Yeah.
So probably is a unique mechanism. They don't dive too deeply, but it could be based on that preliminary information. And because they know the similarity of one of them to this class, they also specifically test that resistance mechanism, and it doesn't do as well as it does. It still is resistant, I would say, but maybe just a little bit better.
Yeah. And I think that
this is I almost think that it actually could be the same thing. It could just be the mechanism of action of fluoroquinolone resistance, just slightly tweaked.
Right, yeah. So that definitely could, because again from the structure we can kind of predict that. And then that brings us to the second to last figure, which is figure 6SG, which it basically finds that these antibiotics are bactericidal in nature through similar ways that we found before. And
so good. All that is excitement, right? They're kind of building us up to say, look, now we did it on a really big library, and we can find some of the same indications that there's a novel antibiotic in the mix.
Yeah, and because I think that for this paper they need to find more than one antibiotic. Otherwise, it wouldn't. Otherwise, all they've done is find one new antibiotic. You need to find more than that in order to prove the concept.
Yep, and that's sort of what they do, and then they show us this again. Now you can see all of the data that they looked at, right? Actually, they don't show you all the zinc data. They only give the zinc data predictions over the cutoff. And actually, what's interesting to me is that it's very clear that their cutoff has a bias for certain clusters. Yes. Right? Because those cutoffs are not so evenly dispersed. I think I see a purple dot right in the middle of everything, but other than that, most of the purple is clustered. And so again, this is going back to the point, adding in this Wuxi library into their data set for training might have introduced some sort of bias that they didn't previously have in the model, and maybe would just need a lot more work to try to disperse a little bit.
I mean, I think the great thing about machine learning, they can literally go back to the original version that didn't have the Wuxi library and run that through it to see whether it has any differences or not.
Yeah, and if they do that too, these are some of the things where I'm just I think these would be really. I'm this paper is whetting my appetite for machine, like previously, I guess, like for me, machine learning is like, I understand it can open up all these different things, but reading this paper has really put me down the line of like where the details are,
Mm.
right? Where details do I want to really see from like the application of machine learning to these types of problems that would make me feel confident that we are going down the right path in terms of application. But again, the important thing I think that they point out is that their hits, the top hits after their filtering strategy, those are evenly dispersed over this graph, more or less. And so they do say that that is a nice indicator of the unbiased way, right, in which this algorithm
I
works.
mean,
Uh,
are they? Um,
that's what they say.
but because
You can get clusters upward, and none of it's in the Ruchi library at
yeah,
all, right? In terms of chemicals.
there's literally one that's quite close in the middle of the Wuchi library. The rest of them are like kind of clustered towards a different area. So, but then again, you wouldn't because I mean, not every structure is going to be antibiotic, so it's going to be it's difficult.
Yeah, there could be an inherent bias in the map of all true antibiotics. So again, I like this map. It's a great one to end off on and just leave on people's minds, right? Because this is the problem that we're dealing with, right? Is that here are all these dots, and there's even more, right? There's a billion. There's like the 0.9 is only 1,000 out of a billion, right? There's way more dots out there in the world, right? How do you know what is an antibiotic? Right? And how do you make sure that the algorithm that you make isn't just like zoning in on something that it's seen in a sector, right? Because even though that sector may have tons of antibiotics, there may be another sector in another place very far away, and how do we find it, right? That's the challenge, and that's what this paper sort of says is trying to urge us to think of machine learning as that tool to crack that nut.
I mean, the thing that's interesting about this paper is how it comes straight from the chemical structure itself, as if there's some intrinsic chemical thing that makes something antibiotic, but it's to do with the metabolism of the bacteria and what it's going to be hitting, and we can't necessarily. So again, we're still doing that kind of random shotgun like approach of trying to hit random things, but then at this point it's using that hits to direct further investigations, find some things that are very similar to that hit or that have similar properties, but it's still
Mm-hmm.
so I mean, again, if you don't hit, if none of these chemicals in that initial learning like training set hit like a area of the bacterial metabolism, then that area isn't going to be covered in subsequent searches that it does.
For sure. For
So
sure. I think actually what you said there is a really interesting thought, and probably in the field, right? People would say, well, this is only taking information from the chemical. What if somehow we were able to integrate the data set, right, to say we can now annotate those chemicals with another functional thing, which is where they might bind to in a bacteria,
yeah,
right? Again, we're not really there yet, right? Like that information doesn't exist, but like perhaps that could also inform assay design. Like maybe they only use one assay to feed into the machine learning
mm.
algorithm, right? Like they use chemical structure and then they use the assay of is it an antibiotic or not. But what if they also fed in, right? Does it bind to bacteria at all? Right? Did it do anything to it? Like does it make a metabolic change in a bacteria? What if they did RNA? I mean, this sounds crazy now, but like we've talked about barcodes before, right? But what if they do like a barcoding approach, right? Where they look at transcripts from bacteria that have been treated with all the different right
Right.
with their training set, and they say here's the way that these different molecules impact the transcriptomics, right? And then they use that to train the model.
I mean,
That plus antibiotics.
that sounds like that'd be amazing. Actually, that'd be a lot of data to chew through.
So
But
more data, yeah.
but
But maybe we don't like. I always think transcriptomics is really just taking marker genes to the maximum. Maybe there's just marker genes that actually have a big impact, right? Like there are stress responses. We know something about bacterial stress response, right? There are stress responses that we can measure in bacteria, and we could use that as part of the training set as well, right? Just what molecules make bacteria stressed.
but I mean, that's not to take away from what they've done here, because they've actually like added a lot more functionality to that the strategy of finding new molecules, and I think that's quite important.
Absolutely. I mean, all of this is just like I'm excited, right?
Yeah,
Like I loved reading. I thought this paper did contribute something big,
yeah,
but a big part of for me, a big part of absorbing literature, right, is also making sure that you don't overstate what the paper is telling
right,
you,
yeah,
right? From the title, deep learning. What is it? Deep learning and antibiotics. A deep learning approach to antibiotic discovery. From that title, I think that it can get overblown. We said it was a top cited thing. It's really hot, right? Like, I just want to make sure that people say, like, this is an approach,
mm,
right?
yeah,
A single approach. And it's really helping me as a more biologist, not a computational biologist, right? Start to imagine the ways in which this tool could come into use, right? And importantly, the limitations of this tool as more and more people start using it, right? Is like a really important thing to keep in mind. And all of these ideas that I have had and I'm saying, they only came to me after I read the paper,
yeah.
right? These aren't things that from before I read the paper, right? This is me taking what I learned from the paper and saying, well, what about this? What about this? What about this? And that's science, right? Like science is all about the questions you ask afterwards.
Yeah, I think one of the most interesting things that this paper could do is teach microbiologists about AI. I think that's uh, a really underrated thing about when you publish a paper that's cross-disciplinary. You actually bring in a lot of new information to people who might not have been exposed to it. Um,
Mm-hmm. Mm-hmm. Totally. Yeah. And like from that, like new disciplines could be formed, new collaborations, right? Like new people working together. Right. This is like encouraging all the microbiologists out there to, talk to the person in the computer science department who's been training things on word data, right? Like they may have abstract principles that work in your scenario.
yeah, I think that so I can so I'm quite glad we picked this paper because it's quite a consequential paper. It's quite this is the kind of thing that is very big for people in the field, and yeah,
Yeah.
because of
In respective fields, right? In the
yeah,
field of computer science, in the field of antibiotic discovery, right? It does a really good job of like putting them together in this like intriguing way that shows application, right? And shows the way that you could get to these pieces of information.
yeah. So I think that's a good note to leave it on. Uh,
Absolutely. Um, Join us next week for our news week where we will survey papers to find something to cover in detail the following week. Probably SARS-CoV-2. I'm not sure.
yeah,
We'll see where we
well,
go there.
we're opening up now, so we'll be covering both SARS-CoV-2 and other papers that catch your eye. And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible we didn't get everything right. I mean, especially there's been so much deep learning in this. I know that I've been at sea for a lot of this, so Danny's been quite good at explaining things to me. But yeah, science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
And you can probably even see some of the biases in what I learned because I try to leave breadcrumbs in the Zotero folders, right, of these different things. Yeah, I totally agree with that. Like we have blind spots as well. You can reach out to us over Twitter with the hashtag #MicroTWJC. And again, as I said before, this is a process peer review, right? Like and absorbing journal articles. So if you had a good time listening to us ramble about microbiology, if it encouraged you to read the paper yourself, then let us know in the comments or let us know what you want us to cover next.
Yeah, for sure. The comments, you can reach us over Twitter through MicroTWC or our respective Twitters, which are listed underneath our faces. And if you have anything unclear that you want to discuss, we can bring up to the next session. It's been a real pleasure chatting with you, Danny.
Same here, Faz.
So tune in next week for more microbiology content.